Today I'm going to talk to you a little bit about CRISPR screening and how we use this in the context of looking at venoms and how they function. So I'm from the University of Sydney in the Dr. John and Anne Chong Lab for Functional Genomics. I don't think I need to give much of an introduction to this audience on venoms. These animals that have developed venoms and venom apparatuses are some of the most feared and revered throughout our cultural psyche, history, and into sort of pop culture today. The evolution of venoms is something that's really exciting to look into just because it's arrived so many independent times and across all of these different species and taxa. So today I'm going to focus on one particular story, and then I'm going to introduce some of the stuff that I'm working on later in that. And I'm going to focus on the snakes at the start. So if you are familiar with this snake, this is the red skin cobra, Naya pallida. Play this video. There we go. And as you can see here, they can spit their venom with quite great accuracy at human faces like this one. And here is our collaborator from the UK, you may be familiar with. And as you can see here, he probably should be wearing maybe a bit more PPE. But these are some beautiful creatures. But the reason we focus on them is because of their medical relevance. So these speeding cobras are generally found in sub-Saharan Africa and then also across South Asia. And you're probably familiar with these snake bite and snake envenomation events is a neglected tropical disease and still something of importance to this day. And especially in these areas, which are a little bit poorer, where they lack these sort of medical facilities that places such as here in Australia we have. And that's important because our current treatments for snake bite, snake envenomation is, of course, antivenom. It's something that is quite expensive to produce and also relies on a cold chain, so refrigeration, which in some places is just inaccessible. And also needs to be delivered in a hospital or clinical environment. And one of the other factors is that because they're made up, these antivenoms are made up of large antibodies, they are very good at treating the systemic effects of envenomation, such as heart or paralysis. But in the case of these spinning cobras, they have a very necrotic venom, and these antivenoms aren't able to penetrate into those peripheral tissues. So to tackle sort of trying to find new treatments and antidotes for envenomation, we wanted to go back to the basics and look at how does the venom and the venom components actually interact with the cell. And in order to do that, we use CRISPR. So to sort of run through with you, if you're unfamiliar how CRISPR works, it consists of a protein, this Cas9, and you can basically give it this guide RNA, and that will tell it exactly where you want it to go into the human genome, and it will go there and make a cut. Your own DNA will then try and repair itself, but usually it doesn't do a very good job, and therefore that gene is then misaligned and will be essentially deactivated. So we call this a CRISPR knockout. So you can see if we take a cell and we knock out a gene, we can then study how it functions when it's not in the cell anymore. You can do this with one gene, but we do this across the whole human genome for every coding gene. So that's around 18,000 genes, and we also use 4 guides per gene, just so that there is a redundancy in the system. So put it this way, so we have our guide library, we clone it into antiviruses and then package it into a large pool of cells. And at this point here, we select those cells. And I kind of like to think of this part here as kind of like a battle royale. So we add the venoms there. So if you're familiar with the Hunger Games, That large pool of cells, each of these cells have one different gene missing, and we want to see which genes will survive that venom onslaught. So I'm sorry if I'm going to spoil a 10-year-old film, but we look at the survivors. So here, the surviving cells versus like the initial population. Once we've added the venoms, we extract their DNA, sequence them, and then see which genes actually helped, when they're not there in the cell, prevented the venom from working. So we did this with, I'll bring you back to the red spitting cobra and also the black neck spitting cobra. And what was really interesting was that we found similar genes to be implicated in their action. So when these genes are not in the cell, the cell actually survived better. So when we take this through pathway analysis, we found that these genes related to proteoglycan biosynthesis. And as you can see here, I've sort of illustrated those top hits. So this kind of made sense to us because these proteoglycans, they're like long sugar chains on the cell surface within the extracellular matrix, and they generally function as ligands for different things. So we kind of think that this is kind of like a little door handle into the cell that the venom has evolved to latch onto and then initiate any sort of cytotoxic action. So then you can visualize if you don't no longer have that door handle on the cell, you can't get in anymore. And we found it interesting that this was across the two different cobra species. So the next thing we thought was, how do we use this information to find a treatment? And luckily, there are drugs on the market that are FDA approved that are actually analogs of this proteoglycan structure. So what we wanted to do was see if we can flood the system, so give a lot of this drug after an envenomation event, and then maybe it will coat all of that venom and protect the cells. So this is just an in vitro assay. Here you have the cells with just the snake venom, and you can see that they is quite cytotoxic. But when we add the drug, they remain in this nice monolayer. They're quite nice and healthy. And we found this with our drug and also some low molecular weight variants as well. Now, this is just the whole venom, but of course we know the venom is full of lots of different things. So I wanted to see maybe if the drug was acting on a particular component of the venom. So I just did a fractionation of these cobra venoms into their known major components, and we did a surface plasmon resonance experiment. So here we attach our drugs to the flow cell in the chamber, and then we flow all of these different fractions over it, and then see how well they bind. So all of these here, we can see much binding. We did come across good binding with these basic PLA2s and these 3-finger cytotoxins, 3 and 4. And then I took these fractions then back into my cell assay with the drug, and we saw that the cytotoxin 1, this is a 3-finger toxin, is still quite cytotoxic, but the drug wasn't that great at reversing that. The basic PLAs in this context weren't that cytotoxic. But these 3-finger toxins were both cytotoxic, and it appeared that the drug was counteracting that action. So we're quite certain, or this made us, all of this data combined with the binding, and this might be quite specific to this. So then going back to look at some other snake species, just to see how universal this might be. So these are some South Asian cobras, spitting and non-spitting, and we know that they are also quite, there's a lot of these 3-finger cytotoxins 3 and 4 in their composition, and our drug were able to rescue those. But if we look at other species, so these vipers, which don't have the 3-finger toxins in their venom, our drug didn't have any effect on those. So just another confirmation there. And obviously, when we're looking to try and find these new treatments, we do want to look here in these specific venom components that are highly toxic and also abundant. And as you may know, there is already some inhibitors for the snake venom metalloproteases and PLA2s, but we don't currently have a three-finger toxin inhibitor, so this might be of interest. We also did the in vivo assay for this. So here we just dose the animal on the flank with the venom, then add our drugs both intradermally and subcutaneously, and then measure the necrosis. So the subcutaneous injection, we really wanted that to see if that would work, because this is already how the drug is currently approved to be used by the FDA. And we do see a reduction in the lesion area with that. So somewhat, tweaks to be made to this assay, but it's a good first step. So kind of showing you here how we can go from just a single venom, not know all that much about how it works on the cells, run a CRISPR screen, and then identify a pathway and a possible mechanism, and then use that information to find potential therapeutics. So this is kind of the pipeline for our lab, and if you're interested in looking at your venom in this context, you feel free to talk to me about that. But obviously, this is just one animal. We look at a whole range of different things in the lab with a lot of different collaborators. And one of the things that I found really interesting recently is we've done also CRISPR screens on this jellyfish here, the Atlantic sea nettle, and also a jellyfish from Australian waters, the blue bottle. And I'm sorry, this is a little bit messy, but a lot of those same gene targets that we found in the snakes, we actually found in these jellyfish as well. And that was something really exciting because, these are, going back to the evolution of it, things that evolved, hashed out over 60 million years ago, but they might have converged on a common pathway to attack cells. So we found a few different common pathways now, and we're hoping that with this information, we could come up with some universal treatments in the future. And so with that, I'd just like to thank my lab here at the Charles Perkins Centre and also all of the collaborators on this project. Thank you.
Thank you very much, Tian. Are there any questions from the audience? While we are waiting, I will start with a question. So you talked about the glycans. Do you know if those genes are also affecting the serine protease glycosylations? Or are there any serine proteases in the venoms you investigated?
I feel like for these spitting cobras, the main components are those three-finger toxins and the PLA2s. So I don't think, and maybe there is serine protease in there, but a very small amount. So it probably wasn't enough there to pick it up. It would we'd want to run a CRISPR screen with something that has that more enriched to see.
Okay. And one question, I'm not familiar with this kind of tests from the drugs directly on the cells. Would it be possible to apply the drug also directly in the wound? Or is it always necessary to go over the systemic way by indermal or directly into the blood system?
Yes, so the intradermal and the subcutaneous is at the bite site. So we'll just, it's injected under the skin. So I think we want that treatment route so it doesn't have to be done intravenously around the body. I think we're sort of envisioning for snake bite, at least it could be a package as sort of like an EpiPen kind of a system, so that it can be at the bite site, as I said before, so that it doesn't have to go all the way through the body. So yes.
But would it be possible to make some kind of drops that you just drop on the wound, or would you always try to prefer the inhibition that not further tissue will be damaged?
Yeah. So are you saying that you could put it on as like protection before? Yeah, I think that's possible. We are looking at for the jellyfish in particular to possibly formulate it as like a cream so that it could be applied beforehand. But we wanted to go the injection route first just because that's how it's used currently. So for translation, it could be faster.
Super interesting. I like the idea of the cream against the jellyfish because it's a total. There's nothing like this. Oh,
I think all the creams at the moment on the market are just like antihistamine, or some sort of topical painkiller, but nothing that seems to be potentially like actually inhibiting the toxin itself.
yeah.
So yeah, that would be something to look into. But I'm not a pharmacologist, so I don't know how to formulate any of that.
That's a really interesting idea. Any further questions?
It's well, it's this about the jellyfish. I never heard about it. And the idea to make, if I understood correctly, so the idea is to have a cream that will prevent from actually, if there is a contact with the toxin, that there is a reaction. But in a case of having the toxin inside the eyes, how is it treated? And do you have also some idea with all the open parts of the?
Yeah, that's a good question. I actually don't know what the current treatment for, yeah, because with the cobras, they do attack the eyes a lot. So I don't know what the current best practice for that is. I feel like if the drug could be formulated as some sort of, eye drop or something, like it would be able to also disperse in that way. So that's a possible avenue for the treatment as well. Yes, I think at the moment, because we've just we're at the stage of this is exciting, this is how we'll do it. Like I don't know all the endless possibilities there could be with how to
of course, it's great. It sounds super cool to make the. Thank you.
Yeah, but yeah, I definitely thank you. Yeah, I really hope that once it's published, because we're getting it published soon, the clinicians can utilize the information. Yeah.